Hey y'all, welcome back to my channel. My name is Jeffrey. In this video, I'm going to discuss both purine and pyrimidine synthesis. So this is going to be a very heavy video. Let's dive right in. There are going to be two main precursors for purine synthesis. Okay, so I'm going to start with purine synthesis here. And you're going to need two different things. You're going to need one amino acid and one tetrahydrofolic acid. Okay, and that's going to be THF. And your amino acid, it can be glycine, aspartate, or glutamine. Those three things, I tend to remember them AGG. All right, and now for synthesis of tetrahydrofolate, you're going to, for eukaryotes, which is us as humans, we're going to receive it from our diet. So we're going to receive it in the form of vitamin B9. It's going to be turned into folic acid in our gut, in our stomach, and then we're going to use this enzyme called dihydrofolate reductase, which is going to reduce dihydrofolic acid or folic acid into dihydrofolic acid and then tetrahydrofolic acid. Okay, and that's going to be inhibited by a drug, and it's going to be called methotrexate, and this is going to be an anti-cancer drug. Okay, and for prokaryotes, for bacteria, they can actually synthesize folic acid without the need of getting it from our diet, and they can do so by making this product called PABA, which is para-aminobenzoic acid, and it's going to be inhibited by sulfonamide or sulfa drugs. So if we look here, trimethoprim is going to be the inhibitor of the prokaryotic form of dihydrofolate reductase. Okay, and what I wrote right here is if you don't have enough folate, if you don't receive enough folate in your system, and you can't produce that tetrahydrofolate, then you're going to have this disease, and it's going to be called macrocytic anemia. All right, so now that we've talked about the two precursors for purine synthesis, let's go into de novo synthesis of our purine, okay? And our base purine is going to be IMP, it's going to be inosine monophosphate. So essentially we're going to take ribose phosphate, we're going to phosphorylate it, and it's going to become phosphoribosol pyrophosphate, and we're going to also use the enzyme PRPP synthetase, okay? So we're going to synthesize PRPP, and then that's going to help feed forward the next reaction, which is with this enzyme, it's going to be called phosphoribosol aminotransferase, and you're going to need a glutamine with that as well, and you're going to form phosphoribosylamine, and then it's going to be turned into inosine monophosphate. Okay, and this can be inhibited by these different types of molecules. So thiopurine, azathioprine, and mercaptopurine. Okay, so these products can inhibit synthesis of our A and G purines. All right, and what's special about mercaptopurine is that it can also be inactivated through this enzyme. It's going to be called TPMT, which is thiopurine methyltransferase. So it's going to transfer a methyl group over onto the mercaptopurine, and it's going to inactivate it. But if this enzyme is inactive, or you have a deficiency for this enzyme, and it can be inherited, you're going to have a buildup of mercaptopurine, and that might have extremely terrible effects for you, as you might imagine, because you'll now not be able to synthesize IMP, and IMP is going to be very important in your ATP, GTP synthesis, as well as, more importantly, your deoxyribonucleotide synthesis for your DNA. Okay, so let's walk through this. IMP is going to be your inosine monophosphate. It's going to utilize an enzyme called IMP dehydrogenase, and that's going to use either GTP or ATP, and that's going to lead to the formation of either adenosine monophosphate or guanosine monophosphate, And you just have to remember that guanosine is always going to use ATP and adenosine is always going to use GTP. So it's just going to be the two converses of each other. And then you're going to phosphorylate it. You can phosphorylate it all the way down to ATP to be used later on, or in the ADP form or GDP form, in the diphosphate form, that is considered to be a ribonucleotide. And if you use the enzyme ribonucleotide reductase, you can actually reduce it into the deoxyribose form, and that is the form that is for DNA, right? So, in addition to that, if you phosphorylate these all the way down to your triphosphates, they can actually feed forward these the converse reaction. So if you think about it, we want equal numbers of purines and pyrimidines in our system because of Chargaff's rule, right? So um, this is a way of maintaining balance in the system. Okay, so we're going to have our deoxy ADP, deoxy GDP, and that can go into the S phase. Or if it sits around the cell for a long time, we can have that nucleic acid turnover as well as some degradation of those periods. Okay. So in addition to that, if you didn't think that ATP and GTP didn't do enough, the synthesized ATP and GTP can actually feedback inhibit those two enzymes for de novo synthesis. So your PRPP synthetase as well as your phosphoribosome aminotransferase. Okay. So moving on, let's talk about the purine recycling pathway. All right, so this is when you have that extra buildup of purines, or when a cell dies, or if you, for example, maybe ingest some meat and it has some extra purines that you don't need. So, or might not be using at that very moment. So you're 80% of the time when we have excess, we're going to recycle those purines. Now, this is going to be the general reaction, which is going to be your purine base, and you're going to use PRPP and a certain enzyme to help reduce it into the nucleotide. Okay, so for here we have the APRT, which is the adenine phosphoribosole transferase. If you think about it, adenine is going to be the base, phospho is going to be you transferring phosphate over, ribosole is going to be the ribose sugar, right? So you have your adenine, and you're going to transfer a phosphate group as well as a ribose sugar group onto that adenine base and turn it into AMP, adenosine monophosphate, okay? And then for your HGPRT, it's going to be hypoxanthine, guanine, phosphoribosole transferase. So hypoxanthine with PRPP, with HGPRT is going to turn into inosine monophosphate. Guanine is going to do the exact same reaction with this enzyme and turn into guanosine monophosphate. And hypoxanthine, I forgot to mention, is going to be the parent base of that inosine monophosphate molecule, right? So we talk about bases, guanine, adenine, cytosine, thymine, right? Inosine is going to be that quote-unquote base for your IMP. Okay, and if you have Leishman syndrome, that's going to be a autosomal recessive disorder. A X-linked recessive disorder, and that is going to lead to your HGPRT not being functional. And as a result, you might imagine that you have a ton of buildup of your guanine and your hypoxanthine bases, and that's going to lead to uric acid buildup. Okay? And that's actually what I'm going to talk to you about right here, which is the purine degradation pathway. This is where we destroy the purines. Okay, so here we have the three different purines, and you're going to use this enzyme. It's going to be called 5' nucleotidase, right? And that's going to convert it down to its base. Remember, nucleotidase. It's going to convert it down to its nucleotide base. And for when you get to adenosine, it's actually going to be deaminated into inosine before it can be broken down further. Okay, so now in this pathway, now we only have inosine and guanosine. Both of them use the same enzyme, and it's going to be called purine nucleoside phosphorylase. So it's going to be phosphorylated, dephosphorylated, and into hypoxanthine and guanine. And then guanine is going to go through reaction with guanase, and then hypoxanthine is going to go through an oxidation reaction uh, uh, to turn into xanthine. Okay, and then xanthine is going to use the exact same enzyme once again to turn into uric acid or your monosodium urate. Okay. And if we're just going to talk about the inhibitors real quick, allopurinol is going to be an irreversible inhibitor of xanthine oxidase. So you're going to basically not allow any uric acid formation. Okay, and that's useful for example, when you have gout, where you have a ton of uric acid and you can't excrete it all in a timely fashion, right? And that's going to lead to uric acid buildup, um, which is very painful. And it's actually a lot easier to excrete hypoxanthine and xanthine from your system than to turn it into uric acid, break it all the way down to uric acid. So that's why we prescribe allopurinol. And then if we have a adenosine deaminase deficiency, then that's going to be called adenosine deaminase deficiency. And that is going to lead to a toxic buildup of what you imagine is ATP, right? Deoxy ATP, because you can't break it down. And as a result, that's going to inhibit your ribonucleotide reductase. If you remember, ribonucleotide reductase is the thing that actually makes those deoxyribonucleotides, right? So you're going to inhibit that enzyme, and that's going to lead to decreased amounts of your T and B cells, which is going to be an immunodeficiency, right? And you can try to treat this through 3 different treatments, and it's going to be enzyme replacement, bone marrow transplant, or gene therapy. And gene therapy is actually a recent type of treatment that has shown some promise. Related to adenosine deaminase deficiency is your PNP deficiency, which is your purine nucleotide phosphatase deficiency. Okay. In addition, certain factors that may influence the buildup of uric acid, even if you live a healthy lifestyle and you don't have these types of uh, um, all your enzymes are healthy. If you intake an excessive amount of meat or beer, these have a ton of these purine bases, and as a result, um, they actually can be directly converted into uric acid after you eat them. So that's going to lead to an abundance of uric acid in your system, and for many people, they can't excrete it quickly enough. So as a result, you can have gout. Um, so in addition to that, if you have if you have decreased amounts of excretion or under excretion, that can also be detrimental in leading to gout. Okay, and you can treat that with this probenecid type of drug. All right. So that's going to be your entire purine synthesis degradation pathway. Now let's move on to pyrimidine synthesis. Okay, so pyrimidine synthesis is going to use also 2 different things. So it's either going to use aspartate, glutamine, and carbon dioxide, as well as THF, which is tetrahydrofolate that we talked about before. Okay, so carbon dioxide in your system is going to be turned into bicarbonate, and you're going to combine it with glutamine with this enzyme called CPS2, and that's going to turn it into carbonyl phosphate, which is going to go through a series of reactions to turn into orotate. And orotate is going to be turned into OMP through orotate phosphoribosyltransferase transferase and PRPP, like we talked about before. It's going to be decarboxylated by OMP decarboxylase into UMP, and then UMP is going to be phosphorylated into UDP. Okay, and then UDP can be phosphorylated again into UTP. Okay, and remember, uracil is going to be that for RNA only, right? And increased amounts of UTP can actually be a reversible inhibitor or feedback inhibit your CPS2. In addition to that, the OPRT, OMP decarboxylase, both of those enzymes basically form what is called UMP synthase, because if you look at it, you need both of these to synthesize UMP, so UMP synthase. And then you can, if either of these enzymes are going to be dysfunctional, it's going to lead to erotic aciduria. Okay, and that basically just means either one of these enzymes are deficient, you're going to have a buildup of orotate in your system, and as a result, you're going to have lowered amounts of red blood cells, as well as you can treat it with uridine administration. Okay, so uridine administration basically just means that you're administering the uracil already, and you're not going through this pathway, so you can actually move on. And that's what you want, right? Because you want to be able to synthesize your deoxycytosines and your deoxythymidines, okay? In addition to that, ATP and PRPP help boost the activity of your CPS2. Okay, so once you have your UTP, you use an enzyme called CTP synthetase, which is going to synthesize your cytosine triphosphate, and then you're going to actually dephosphorylate it. So you're going to use a phosphatase, remove a phosphate group, and you're going to get CDP, because remember, your ribonucleotides have to be in the diphosphate form, okay? So if you just use ribonucleotide reductase, you can directly turn that into deoxy-CDP. Okay? But there's actually a very complex pathway to turn it into deoxy-TNP, and that's going to be your thymidine. Okay, so when you have your UDP, it can also undergo that ribonucleotide reductase reaction, along with, remember, you have to have that thioredoxin with the NADPH to help facilitate that process. And you're going to turn the deoxy-UDP into deoxy-UMP, and then you're going to remember THF is used in every single pathway here. So for both purines and pyrimidines. So it shows up again. You're going to have your THF, which is going to be synthesized by that dihydrofolate reductase, remember? And you're going to add in a methylene group, turn it into methylene THF, and help facilitate that reaction. So you have your thymidylate synthase, which is going to synthesize your thymidine, okay? But yeah, so essentially like this entire reaction right here donates one carbon. Remember, methyltrexate is the inhibitor of dihydrofolate reductase, and this is going to be the eukaryotic inhibitor that we talked about before right here, okay? And then 5-fluorouracil is also going to inhibit your thymidine synthase, because you're going to inhibit the enzyme that actually synthesizes it. All right, so that's eventually going to lead to your deoxy TMP, and that's going to go to your S phase. All right, so I hope that this video has been helpful. There's not actually, there is a pyrimidine degradation pathway, but since there's no diseases associated with it, we're not going to talk about it because it's not clinically relevant, okay? So I hope that this video has been helpful. It's been a little rushed, and just leave any comments if you have any questions, and have a great day.